So in this lesson, we're going to work through a past paper question. So this is from the winter 2013 session. It's paper 62, it's question 1. So this is one full question out of an alternative to crack paper. So you will notice that this is from before your syllabus change, but it's okay, the content is still the same. There were some tweaks made to the content, but if you use those old papers wisely, they are still a very valuable resource. So what we're going to do, we're going to work through the whole question. We're going to pay particular attention to where we see things relating to our variables, because we were talking about independent variables, dependent variables, and controlled variables in our last alternative to crack lesson. So let's get started. Okay, so catalase is a common enzyme found in both plants and animals. So as soon as we see the word enzyme, we should be thinking of temperature, pH, substrate concentration, enzyme concentration, these things that affect our enzymes. Okay, some students investigated the activity of catalase in seeds and seedlings. Did you catch it? Investigated the activity of catalase. So is catalase our independent or our dependent? Okay, so we need to keep this in mind. What is our independent? What is our dependent? They used extracts from soaked seeds and from seedlings which have been grown for 4 days. All the seeds and seedlings were from the same plant. Same plant. So we've controlled here. Catalase breaks down hydrogen peroxide into water and oxygen. And there is our formula. So you might not know this one yet, but you know that this one is water and this one is oxygen. So this one must be your hydrogen peroxide, H2O2. It's something you're going to come across again and again. So it's worthwhile being familiar with it so it's not so foreign when you see it. Okay, so catalase is breaking down. So we're starting off with hydrogen peroxide, we're ending up with water and oxygen. Remember, this is O2, it's oxygen gas. So figure 1.1 shows the apparatus used to compare the catalase activity of the 2 extracts. We are comparing activity of 2 extracts, okay. So comparing activity, this sounds like something we're going to measure. And if we're going to measure it, it's going to be dependent. If it's something that we're changing on purpose, something that we're setting, the 2 extracts, this is going to be our independent. And whenever you read an alternative crack paper, this is what you should be doing. You should be working through nicely. Okay, so this was done by counting the number of bubbles of oxygen released in 1 minute. Okay, counting, measuring. So that is how we are measuring our dependent variable. Oxygen starts to be released as soon as hydrogen peroxide is added to the extract. Now, they say as soon as, and it is like crazy fast. It is literally as soon as they touch, it's immediate and it goes whoosh, and that oxygen is produced. Okay, so as soon as, they are not joking. It is as soon as. Okay, now think about it, right? There's a bung here, okay? So let's just work through figure 1.1 a bit. Sorry, let me slow down. So in our test tube here on the left, we've got the extract. So that has our catalase in it and hydrogen peroxide solution. So here they are mixed. Enzyme and substrate. They're producing foam. This is your O2 bubbles. It's creating this foam. But you've also got oxygen coming out through the delivery tube. So then you can see the bubbles that are coming out here. So we can't count bubbles in something in air. We need to see them in water. So we deliver the bubbles all the way through and it bubbles out this end. Okay, so the thing to remember is that when the rate of reaction is very high, the rate of bubble production is very high, which means that you've got to count 1, 2, 3, 4, 5, it's very difficult to count those bubbles. Okay, so keep that in the back of your mind, because remember we talked about errors as well, and accuracy and all of those sorts of things about the experiment too. So you've got to keep in mind where could the problems be? So counting bubbles could be a problem. Also, who says that every bubble is the same size? Okay, there's another level of inaccuracy coming in there through our experimental design. Right, so what happens? How does this work? 2 grams of extract from soap seeds was used. Okay, we like that because this is precise, it is accurate, and it is controlled. Okay, it was placed in a test tube labeled seeds 1 as shown in figure 1.1. So there's the extract that they're talking about. Hydrogen peroxide was poured into the test tube. The bung was quickly replaced into the top of this test tube, but quickly is not as fast as soon as. So we've actually lost some oxygen because it can escape between that time that the hydrogen peroxide is poured in and the bung is placed. So if you're very quick, you lose less. If you're not so quick, you lose more. So we're getting errors coming through in our experiment. So the number of bubbles of oxygen released in 1 minute was counted and recorded in figure 1.1. So these are measurements of our dependent variable. Okay, another question that comes up here. We had 2 grams of extract from soaked seeds. How much hydrogen peroxide was poured in? Did we just slosh it in? Or did we measure 2 moles or 5 moles? Because remember, if we want to be able to repeat this experiment, we must have precise instructions. Otherwise, it's not necessarily the same. Okay, so this was repeated with another extract of soaked seeds. Repetition. We like that because when we have repetition, we increase reliability of our results. Okay, results were recorded in table 1.1. The whole procedure was repeated with extracts from 4-day-old seedlings, remember from the same plant, labeled seedlings 1 and seedlings 2. So we have repetition for the seedlings again. The results were recorded in table 1.1. So let's go have a look at this magical table 1.1. Here it is. So we have the extract, we have seeds 1 and seeds 2. So here we have our repetition, if we can spell repetition. And that is going to improve our reliability. Remember, we also said whenever we look at our table, we're always going to have our independent variable on the left and measurements of the dependent variable on the right. So the first thing they asked us to do is to describe the results. Well, here is actually a very nice indication of why we want to do repetition to increase reliability. These answers are not the same. Okay, you can see that they're closer. So seeds are closer to each other, seedlings are closer to each other, but it's not the same exact measurement. So again, it's sort of letting you know that we've got inaccuracies within our method. Okay, so if we're going to describe the results, we're going to do it in bullet points today in the sake of time and actually wanting to get through the entire question. So the first thing, the bigger one, okay, the biggest one, most obvious one, is that seeds have a higher number of bubbles released in 1 minute than seedlings. Okay, so now bubbles of oxygen released in 1 minute. You'd write it out in full if you're writing out your full answer to this. Okay, so other one is that within this, we've got 3 things that we need to describe. Okay, so we've described the overall pattern. Now we need to describe about seeds and we need to describe about seedlings. So we can say that seeds 2 is greater than seeds 1. And the same thing for seedlings 2. Okay, and remember also when we're describing, we like to give values. So 50 versus 43 and 37 versus 30. Now, I am assuming that you can turn these into beautiful sentences with all the words that you need. Okay, these are our three main points that we want to talk about. And the other thing is you see here now you've got a pattern also that 2 is bigger than 1. 2 is bigger than 1. So does that mean that when you did the second experiment, when you did the experiment the second time, you were better at doing it? Okay, so remember that can also have an effect. You get, when you do an experiment and you do it again and again and again, then you get better at doing the experiment. So maybe your time to get that bung in gets shorter and shorter. So you're actually catching more of the oxygen for measurement. So you're not losing so much. So just keep all these sort of things in the back of your head. Okay, state the conclusion that can be made from these results. Okay, so we don't want to talk about now within seeds and within seedlings for here. For a conclusion, we want to look at what is happening, what is the difference that our independent variable makes. So our conclusion here relates to the independent variable. So our independent variable here is the source of our extract, seeds or seedlings. So we want to then say that if we have more oxygen bubbles produced, then we have more activity of enzyme. So catalase is more active in seeds than seedlings. And we can compare this directly because in this experiment, we have controlled these seeds and seedlings are from the same plant. So we can make a comparison like this. If they were from different plants, we wouldn't know if there's a difference between seeds and seedlings. But because we've controlled that variable, we can then make a comparison with them. That is the end of part A, describing the results. So now you've got a good idea of what your experiment is and how to describe the straightforward results. If we move on now to part B, we are talking about error. So state 2 possible sources of experimental error in this investigation. We've got a few things to think about. Remember at the beginning we said we're talking about enzymes here? Okay, and whenever we talk about enzymes, we think about pH, we think about temperature. Okay, we didn't control those at all. Okay, but we've also got this problem of as soon as. So in this investigation, a particular problem is how fast did our bung go in? We've also got the problem of bubbles are not a standard size. So, I mean, if we count 10 big bubbles and 50 small bubbles, which one is more oxygen? And we've got that problem where we said we just pouring in hydrogen peroxide. Now, that's not very accurate. I mean, if we put more hydrogen peroxide in, we're putting more substrate in. So we need to actually control those variables in order to be able to see the effect of that independent variable. So let's go for ones. I mean, we haven't made any mention of temperature and pH here, but we have got a couple of problems directly related to the experimental setup. So let's focus on those ones first. So the big one was the volume of hydrogen peroxide is not stated. And therefore, it could vary. And that's not going to give us an accurate result, okay? The other one was oxygen bubbles are not the same size, or they come too fast to count. So you can pick which one you want to use, okay? So, suggest how to improve the method to reduce one of those errors stated in B part 1. Woo, now, okay, so the other one, let's just make a note here also. We wanted temperature and pH to be controlled because we're working with an enzyme. So let's think about it. You know, the volume of hydrogen peroxide, okay, we're going to be a little bit naughty. They're going to want 1. So in the exam, you're only going to give 1. For now, because we're using this as a revision, then we're going to talk about everybody. So volume of hydrogen peroxide, we could do exact volume of H2O2, measure with syringe. Okay, if we're talking about oxygen, how we actually accurately measure this oxygen, we could look at the volume of oxygen produced instead of bubbles. Here we would use a measuring cylinder to trap the gas. So then you can read the gas from the top of the measuring cylinder, and then you can see how much in displacement and all of that fun and games. So if you're not so sure about that one, go read up on it. Okay, how do we control temperature? We would use a thermostatically controlled water bath. How do we control pH? We would use a pH buffer. Okay, so out of all of these, I think I would probably go for the measuring oxygen, because what we want to do is we want to think about what's our independent, what's our dependent, what are we really looking at. And in this case, we're looking at production of oxygen as an indicator of the rate of reaction. So what we really want to do is get an accurate measurement of that production of oxygen. So measuring the volume of oxygen rather than counting bubbles is a far more accurate way of really understanding the effect of our independent variables in this case. So again, everybody thinks differently. Next section of this one gets on to actually doing some experimental work yourself. Okay, so remember we had foam produced because the oxygen is being created. Okay, and after the reaction had finished, the 4 test tubes had different heights of foam. Here are the 4 test tubes, seeds 1, seeds 2, seedlings 1, and seedlings 2. There's the extract and the peroxide at the bottom. There's a foam with all hundreds and thousands of little dots. And they even show us here how the height of the foam is measured, from the surface of the liquid to the very top of the foam. Okay, because remember, whenever we control something, whenever we measure it, we want to be consistent. So if we were going to measure to the bottom of the foam, it's quite different to if we measure to the top. So if we measure to the top on one, we must measure to the top on all of them. So what do they ask us to do next? They asked us to measure the height of the foam in each of the test tubes shown in figure 1.2 and record in table 1.2. So you've got to measure from this height of the liquid to the top of the foam for each of these test tubes. If you look at figure 1.2, there's our extract, which is our independent variable. The height of the foam, what we're measuring, is a dependent variable. They tell us measure in millimeters, so make sure you do that. Remember, you're generally measuring in millimeters when it comes to biology. So you put on your ruler, and I did print it off and do it on paper. So seeds 1 comes out at 56 millimeters, seeds 2, 67, seedlings 1, 37, and seedlings 2, 42, which is quite nice because it also mirrors those same results that we got from the oxygen count. Seeds have more than seedlings, and 2 has more than 1 in both cases. Also remember, when you're doing a table, your units stay in that heading. They don't go in the blocks with the numbers. Units are only in headings. So we just put the values in the table. Okay, so state the conclusion that can be made from these results. So what is the conclusion that we get? Again, conclusion is going to link to effect of independent variable. So we want to group these two, and we want to group these two. And we can see 56 and 67, both higher than 37 and 42. So we've got beautifully exactly the same pattern as we had before. That catalase is more active in seeds than in seedlings. Makes sense. Okay, so what comes next? They asked us to state and explain whether your conclusion in A part 2 is consistent with your conclusion in C part 2. So A part 2 is all the way here. Catalase is more active than seeds and seedlings. C part 2, catalase is more active in seeds than seedlings. So, are they consistent? They're not only consistent, they're exactly the same. Okay, so yes, they are consistent. Okay, why? We have more bubbles and more foam for greater activity. Both have higher activity for seeds. Okay, again, I'm writing bullet points, little notes. In an exam, you write this into beautiful sentences. And you include all the magic words that you need to in order to make it make sense. Okay, so that is now the end of A part 2. So we've C part 3. I'm reading A part 2 here in the question. So we've now established that when we do it with counting bubbles, when we do it with measuring foam, we get the same answers. So the last part of this experiment question is all about the quality of this experiment and how we could take this further. So we start off in D with explain why the tests for seeds and seedlings were repeated. And we are smiling to ourselves because when we saw at the beginning about the repetition, we said, haha, reliability. Okay, when we increase repetitions, we increase reliability. So we are increasing the reliability of the results. But now that's only 1 point. And there are 2 marks here. So we have to think a little bit further. Why else would we repeat? Okay, so we're going to increase the reliability of the results. But what does that really mean? So if we had results that went 10, 11, 11, 9, 10, 21, 10, 11, that 21 sticks out. Okay, if we'd just done that experiment once and got 21, we would have believed 21 was the answer. But when it's repeated a dozen times, you can see, no, 21 is a weird one. So why do we call that weird result? Okay, in biology, we're going to call that an anomalous result. So repetition allows us to identify anomalous results. Okay, so we repeat to increase reliability, and part of that is identifying anomalous results, because then we would leave that anomaly out of our calculations for averages and things like that. Okay, we know something went wrong in that experiment. Seeds and seedlings were crushed to make the extract. Suggest 2 reasons why whole seeds and seedlings were not used in this investigation. So we need to think about it. Okay, so we have seeds. And we have seedlings with a root and a leaf, something like that. Okay, now remember, if we want to compare a seed against a seedling, we must be comparing the same thing. So obviously they look different, okay? The catalase is, where is it? Let me think, hold on. Catalase is an enzyme. Enzymes are inside cells. So first reason we have to crush is to actually get the catalase out of the cells. Okay. The other thing is, of course, where in the seed, where in the seedling. We don't know any of this information, okay? The seed is a different shape and a size to a different seedling. So when we crush them and we extract the catalase, we can actually make uniform, controlled extracts that we can then directly compare. So an extract provides uniform samples which can be directly compared. Because remember, we want the only difference to be, am I from a seed or am I from a seedling? Not shape or quantity or amount of squishing, anything like that. We want it only to be one difference, because then when there's one difference, we can test it. Okay, right, the last bit goes on again, thinking about now variables, what do we do? Okay, so another group of students wanted to investigate the activity of catalase in different types of seeds. Different types of seeds. Okay, so are you catching it? Did you get it? Here's my independent variable, different types of seeds. So for this investigation, suggest a variable to change. Well, that would have to be your species of seed, because the variable that you change is your independent variable. Two variables to keep constant. What do we need to control? We need to control things like the mass of the extract, the volume, and the concentration of hydrogen peroxide. We're going fast here because we're approaching time. Also, we need to control the time to gather oxygen, same like we did in here. Okay, how long are we going to measure for? And we need to control temperature. And we need to control pH, because we're working with enzymes. So if we want to look at an effect, we have to control everything else. What are we going to measure? Well, we could measure foam height. We could measure bubbles produced, or we could measure volume of oxygen. And what would be a suitable control? Remember, this is now experimental control, a control for your entire experiment. So we would either want boiled extract, because of this course, this is now denatured enzymes, so they're not going to do their job, or water instead of extract. Okay, so we just made it in a sprint through the whole question. Go back and have a think through it. Go back and read the beginning of the question again, and make sure that you're understanding how it all links and why it is so important to read that question thoroughly and think about it from the very moment you start reading it. Okay, you never read for fun in a bio exam. It's always information that you need to take note of.